Good afternoon, and welcome to the latest event in our Lab Notes series. My name is Alex Smith, and I'm from Alzheimer's Research UK. I'm really happy to welcome you here today for what is the 5th event in our series of online events. Lab Notes is an opportunity for dementia researchers to share their work with you and to answer questions that you put to them. This event, along with others in the series, are being recorded, meaning that you don't need to worry about attending every single event, as they will be made available on demand. You might notice that we're using the live subtitles feature in Zoom, which you can turn on and off using the CC button at the bottom of your Zoom window. You may notice that there may be some errors in there, but we're working on it. And it's just an accessibility feature that we plan on improving in the future. So as always, before we get going, we're really interested to know who we're talking to today. And since we can't see you, unfortunately, we have a poll that will appear shortly. This is important for us to find out what motivated you to attend today. So I'll pause for a minute and give you a chance to answer these now. Okay, so hopefully you've all had an opportunity to answer those questions there. So I'll bring up the results. So I'm just looking for them now. So people with a perceived average knowledge of dementia research, and you attended mainly because maybe a friend or a family has dementia, or your work is related to dementia. And a lot of you have attended before, so I'm happy that you felt it necessary or worthwhile to come again. So thank you for joining us once again. And for those of you that are new, welcome along. So I'm just going to share my screen now. Hopefully you can see that. So the plan, if you've been before, you'll know that there's a format to these events, but the plan this morning is a little different than our usual format. But I'll still be giving you a brief introduction to kick things off. And after which we will show you some pre-recorded footage from inside the labs at King's College London. Dr. Bea Gomez Perez-Nievas will introduce us all to the network before handing over to Jodi Parslow, Paula Beltran, and Louisa Johnson, who will all be giving you a unique insight into what goes on behind the scenes in a dementia research lab. As always, we'll conclude the event with a Q&A session, and we'll try and answer as many questions as possible in the time that we have. So here at Alzheimer's Research UK, our vision is a world free from the fear, harm, and heartbreak of dementia. And to achieve this vision, we focus on funding research into the diseases that cause dementia. We support scientists in labs and clinics to carry out studies to understand more about what is happening in the brain in the diseases that cause dementia. Their work is helping us find a way to reduce the risk of the diseases, diagnose them better, and develop effective treatments that offer people with dementia a good quality of life. Now, the amazing research you'll hear about today is only possible thanks to the incredible support we receive from people like you. People choose to support us in loads of different ways, whether you host a bake sale, run a half marathon, give on a regular basis, or make a donation in memory of a loved one. Your support always makes a real difference in bringing forward the day when we can discover a cure. And one really important way in which people choose to support us is with a gift in their will. That's one thing I want to focus on today. So leaving a gift in your will is hugely important, and it's personal and a generous decision to invest long-term in our research. These gifts people leave us in their will come in all shapes and sizes, and everyone makes a huge difference in accelerating our vital research. And in fact, gifts in will support 1/3 of the research we fund. Now, just to put that into perspective, into individual terms, into practical terms almost, I want to touch over a case study. And this is of Katie Foster's father, Tim, who was diagnosed with Alzheimer's disease in 2003 and died in 2007, aged 86. Katie cared for him at home until 2005, when he moved into a care home. Now, as the driver of a gun carrier during the Second World War, he was captured during the Dunkirk retreat in May 1940. He spent 5 years as a prisoner of war before making a dramatic escape and return to England. Now, near the end of his life in the care home, Tim often thought that he was back in the prisoner of war camp. And Katie actually told us in her own words, it was heartbreaking to watch him fade away. A man who used to love chess, difficult crosswords, music and dance, and adored his granddaughters. He gradually disappeared to the point where he thought he was back in the prisoner of war camp. Now, Katie has supported Alzheimer's Research UK for more than 10 years as the leader of the Shropshire fundraising group in memory of her father. And during the first COVID-19 lockdown in 2020, Katie rewrote her own will to leave a percentage of her estate to Alzheimer's Research UK, rather than just leaving a gift of a set amount. And again, in her own words, she said, "It was important to me and my family to put my money where my mouth was, so to speak. Leaving a gift in my will means I'll still be there to support Alzheimer's Research UK, and my commitment might encourage others to do so too. Dementia causes heartbreak to so many families here and across the world. I lost my dear dad long before he died, and it's so important to fund the vital research which will ultimately put an end to such sadness. I've made my legacy to Alzheimer's Research UK as a percentage of my will so that it won't lose value by the time that I die." Now, we are aware that leaving a gift in your will isn't for everyone, and it's a really, really personal decision to make with family, friends, and loved ones to be considered. However, it is vitally important to have a will. Dying without one leaves loved ones facing a difficult and complex decision at a time when they aren't often prepared or equipped to do so. It also ensures that your wishes are met at a time when you can't voice them. And it gives you the peace of mind to know that your affairs are in order at a time when you're able to make those choices objectively. Now, we offer a range of information and support around writing your will. Matters like power of attorney and inheritance tax, as well as details of specialist solicitors. And they're all available on our website. And I believe they're going into the chat now. So if you have any queries around gifts and wills, then please head to the website. Now, back to the event today. We've worked with our research network to bring you this event and others in the Lab Notes online series. Before the pandemic, each centre would organise an annual event to share their work with people in their local area. And so we've worked with them to bring those events online. And at this event, you'll hear from scientists in our King's College London network. They're going to give you an insight into what a day in the lab looks like, showing examples of their experiments and work. And with that said, it's my pleasure to show you their footage from the labs, starting with a talk from Dr. Bea Gomez Perez-Nievas.
Hello, my name is Bea Gomez and I am a research fellow at King's. You are going to see some of the experiments that are carried out in our department to help understanding dementia. So I thought I could give you a small tool to show what we've learned about Alzheimer's disease in particular, thanks to research. Alzheimer's disease is the most common form of dementia, and I am sure that unfortunately you all know someone who is living with dementia, as it affects almost 50 million people worldwide. The pathological manifestations of Alzheimer's disease were described over a century ago. Alois Alzheimer, a German doctor, studied the case of Auguste, a relatively young patient that suffered from a strange form of dementia. After her death, the doctor evaluated her brain and found the presence of what we still call plaques and tangles using silver staining techniques. Plaques are shown here, were abnormal accumulation outside the brain cells, and tangles that are shown here were found within the brain cells. The descriptions that he made were actually pretty accurate, and we know now that Alzheimer's disease is characterized by the presence of extracellular accumulation of a protein that is called amyloid beta, and intracellular accumulation of another protein that is called tau, that in this case gets hyperphosphorylated, that is a modification that proteins can suffer, and accumulates in the neuron. This leads in the end to synapse loss. Synapse is the communication between two nerve cells that helps, for example, form new memories. Then also neurons die, and ultimately even there is brain atrophy. We also know that these pathological hallmarks are accompanied by the presence of inflammation in the brain. It took some years for researchers to analyze the components of those plaques. And research in the field has advanced a lot thanks to generous brain donations from patients, but also healthy subjects. So in the late 80s, researchers could isolate the plaques from brains and the substance that was present in that plaque, and could identify the main component of it as amyloid beta, and also that it came from another protein that is called amyloid precursor protein. In parallel, researchers in the UK actually found out what was the structure of tau protein. This is very important because once the proteins are identified, we can also investigate more in depth what they do. A little bit later, there was a great improvement in research thanks to the advance of genetics. So in our body, the information is coded by genes that determine the expression of proteins like the keratin in the hair and collagen in the skin. Genes are part of the DNA that is made of a sequence of letters that the body machinery reads to translate into proteins. Once A beta and tau proteins were described, the genes that codify for these proteins were also isolated. And several mutations of the genes were found in the populations. These mutations meant that people who carried them had early Alzheimer's disease due to more accumulation of amyloid beta. I wrote here some of the mutations that were found, especially in that amyloid precursor protein. So people with these mutations are more likely to develop Alzheimer's disease earlier in life. Especially mutations that lead to the creation of more A beta that accumulates in the brain. This also, the identification of these proteins and genes also helped researchers to create animal models for research. And this is very important because with animal models, we can test hypotheses. So with human brains, let's say we only have like the final picture of what happened in the disease, but if we have mice and cells, we can see what are the consequences over time or A beta exposure, for example. Or in the case, we can culture nerve cells from mice or even from donors that carry the mutation, and then what can get more insight into what are the mechanisms of A beta mediated toxicity. So this gave us a lot of information in the early 2000s, and led the researchers to postulate one hypothesis that is called the amyloid cascade hypothesis. Was used to explain the disease during many years. According to this hypothesis, a continuous accumulation of A beta in the form of plaques is the central early event in the physiopathology of Alzheimer's disease. This assumes that neurotoxicity of beta amyloid drives the neurodegenerative processes. Although it was very fair to make this hypothesis based on genetic and in vitro and in vivo data, more recently there has been a great advance in imaging techniques, and we can now detect A beta with a non-invasive scan thanks to one ligand that it can be injected into the blood and gets to the brain, and it's not toxic, so that help us detect A beta in the living brain. And we found that researchers found that almost a third of the population actually has amyloid beta in their brains. Also, there was a large study done in non-volunteers, and they found out that relatively high participants of the study show no cognitive decline when they die, but actually their pathology corresponded to Alzheimer's disease in a postmortem exam. So why do these people not get dementia despite having AD pathology in their brains? If we are able to understand how these resilient people, how their brain works, we can maybe discriminate what is really important for dementia and Alzheimer's disease. When I was a postdoc in Boston with the collaboration between several Alzheimer's disease centers, we managed to collect brains from these people who were cognitively normal but had the AD pathology. These are rare cases, so it was a lot of effort, and we call them mismatches because there's actually a mismatch between the clinical and the pathological diagnosis. So if you compare these two brains, the mismatches have the same amount of pathology as the AD patients that we know that show signs of dementia, but they are cognitively normal. In fact, they don't lose neurons, and their brain thickness is preserved. This leads us to conclude that there must be something more than tau and A beta that contribute to dementia in AD. And one of the things that we find is that, interestingly, in these subjects, there was no inflammation reaction to the presence of plaques and tangles. So usually in the AD brain, we can see how glial cells, that are cells responsible for inflammation in the brain, get activated. But this didn't happen in our mismatches. Microglia, which are labeled with this marker here, act as surveillance cells looking for pathogens and eating them up to protect neurons from toxic substances. Astrocytes are very important in giving food to, they're here, they are called astrocytes because they have the star shape. They are very important for giving food to the neurons and remove the garbage that accumulates in the brain. That is very important, the neurons to have a nice environment. However, it seems like in disease, they get activated, and instead of being nice to the neurons, they start to release toxic factors. And in fact, other researchers have found that microglia in particular can start eating up the synapses, the connections between nerve cells, and therefore contributes to cognitive decline in Alzheimer's disease. Astrocytes are always found around the plaques, and as I mentioned, they stop being supportive to the neurons and they start releasing toxic substances. So a lot of attention is now put into these glial cells as a possible mechanism of neurodegeneration in Alzheimer's disease. So just some final remarks to conclude. After years of research, we learned that A beta and tau are necessary to develop AD. If you only have tau mutations, for example, you have different forms of dementia that are called tauopathies. But perhaps targeting just them, or at the moment we are targeting, won't be sufficient, at least in some patients. We know now that AD is a really complicated disease and it's multifactorial disease. We know that inflammation and other cells play an important role. And we know that synapse loss is actually the best correlate of cognitive decline, so it's something that we need to tackle. And the early diagnosis is advancing very fast these days with the discovery of new biomarkers, and that's going to help us identify which patients we need to treat and to treat them early, because perhaps we need to separate, for example, early onset of familiar Alzheimer's disease patients from sporadic Alzheimer's disease. Also, the advances in neuroimaging are helping researchers greatly. So I think we've had a lot of disappointments over the over the years, with the lack of effect of several clinical trials that were targeting amyloid beta, but we've made a lot of progress, and hopefully in the future we will find a more multifactorial treatment of Alzheimer's disease. Thank you very much.
Hi, my name is Jodi, and I'm a neuroscientist and PhD student at King's College London. I'm also a science communicator. As part of my research, I use fruit flies. This is because nearly 75% of the genes that cause diseases in humans are also found in fruit flies. They have short, simple reproduction cycles which can be manipulated by temperature, and so this means that several generations can be observed in a short matter of months. They're relatively simple and inexpensive to maintain. Here you can see me flipping our fly stocks into new food. This keeps the fly stocks healthy and alive for years. Fruit flies are small, less than 3 millimeters long, so we use microscopes to look closely at them. Here I'm showing you the empty pupae on the side of the vial from which the new generation of flies have emerged. This is because the flies mate, they lay eggs in the food. The eggs turn into small larvae, which eat and live in the food, making it mushy. These larvae then grow big, climb up the side of the wall of the vial, where they form pupae, and this is where the adult flies emerge from. To collect these, I insert a fine needle into the vial, which dispenses carbon dioxide to sedate the flies. I'm then able to tip these out onto a porous pad, which also dispenses CO2. Once I have double-checked the genes that I want my flies to have, I'm able to use a fine paintbrush to manipulate and move the flies around to get a good look at them. Here's what I can see down the microscope. Here is a female fly with curly wings, and here is a new fly that has recently emerged from the pupae. As you can see, its wings are neither straight nor curly, they're still closed. Of course, we can also use the microscope to check the general health of the stock and make sure there's no parasites. And we can also have a look at the flies to tell the difference of males and females in order to set up crosses for the flies to breed offspring with a specific set of genes. This can be time consuming as flies are able to generate very quickly. Once I've collected the flies that I'm interested in, I can start my experiments. One example of the experiments we use flies for is the climbing assay. I anaesthetise the flies and I put them in these tubes with 1 centimetre markings on. My flies are first of all tipped and tapped. They're then turned round, tapped gently down to the bottom, and I start a 20-second timer. I watch these flies as they try to climb up the tube and record how far they manage to climb in a 20-second interval. I repeat this 7 times and then take an average of these trials. This test takes advantage of something called negative geotaxis, which is a fancy way of saying that the flies have a natural escape response, whereby they climb upwards after being given a startle. This is a very easy measure of motor ability for my research in motor neuron disease and ALS genes. And if I now show you this video of flies that have been aged and contain ALS genes, you'll see the difference. So this time when I tap down the flies, you'll see that very few of them are able to actually climb up. The majority of them are stuck at the bottom. Importantly, ALS is a disease that affects your motor abilities. As a reminder, here's a picture of a fly in the centre of my hand. They're actually only 3 millimeters long. I'm able to use a microscope to dissect out the fly's brain, which is obviously very small. As well as working with flies, I also work with cells. In particular, I work with human neuroblastoma cells, and they are stored in these special cryogenic tanks which preserve cells at temperatures below minus 150 degrees Celsius using liquid nitrogen. We can thaw the cells when we want to use them, and this is called cell culturing. When we perform cell culture, we have to work in these specialised tissue culture hoods, which have a controlled airflow and make sure that it's a sterile environment. Here you can see me transferring the thawed cells into a falcon tube and then adding in a culture media. This is this pink liquid which contains all the nutrients that the cells need in order to survive and grow. Then I put this cell mixture in a specialised machine called a centrifuge. This spins my samples at a very high speed in order to separate its components by mass. It removes any dead cells or unwanted ingredients from the freezing media. And it leaves me with a nice clean pellet. Here you can see at the bottom, that's where my healthy cells are. I then add in some fresh culture media. Then when I'm ready, I plate these cells into specialised flasks, which are coated so that the cells can stick to the bottom of the plastic. When they're stuck to the bottom of the flask, they're able to grow and expand in number until they're slowly covering the entire bottom of the flask. When they run out of space, they need moving into bigger flasks so that they can grow some more. This is called splitting or passaging cells. In my PhD research, I take healthy human neuroblastoma cells and I treat them with a drug which induces cell death and causes the sick cells to change shape as a way of modelling the way that cell death happens in diseases like Alzheimer's disease. The dying cells then start to release special proteins packaged inside spherical balls which are called vesicles. These vesicles are used as a way for cells to send messages to each other. These flasks of cells are kept in special incubators which mimic physiological conditions such as temperature and percentage of CO2. Here's what the cells look like underneath the microscope when they're healthy and after they've been treated with the drug. As you can see, they've started to change shape and clump. The next part of my research is to collect the vesicle messages. To do this, I use a big version of the centrifuge machine to spin my samples very fast to create a force that is up to 100,000 times the force of gravity. I'm then able to use a specialised equipment that separates proteins by weight, by forcing an electric current through a solution. Here you can see me releasing the gel. I then do something called a transfer, and this pushes the proteins out of the gel and onto a membrane, where I'm able to stain using various antibodies to look for my protein of interest. These proteins may be important in understanding how diseases function. Eventually, I want to be able to collect these special proteins and apply them onto immune cells that exist in our brains and play important roles in diseases such as Alzheimer's disease.
Hi, my name is Louisa and I'm a PhD student here at King's College London. For my PhD project, I'm doing research into Alzheimer's disease and specifically a type of cell in the brain called astrocytes. Astrocytes support the nerve cells in your brain, helping them to do their job. But in Alzheimer's disease, this can go wrong, and this is why I'm interested in researching them. In my research, I do a lot of work looking at cells under the microscope, and so this is what I'm going to show you today. The idea behind these experiments is that I can treat the cells in different ways, for example by adding a drug to them, and then I can look at the cells under the microscope to see if that drug has an effect on the cells compared to when there is no drug added.
Firstly, I grow cells on a plate like this, so each of these little wells has hundreds of cells growing on it. By removing and adding different volumes of different solutions, I can treat the cells and also prepare them for imaging under the microscope. Usually, I will treat the cells with a drug or other compound and then fix the cells. Fixing is where we add a special solution which fixes the cells as they are in the plate, so they're no longer growing and the structure of the cell is locked in place. Next, to be able to visualize the cells under the microscope, we need to make them fluorescent. I use a protocol called immunocytochemistry to make target proteins of interest fluorescent. This uses antibodies which bind really specifically to their target, allowing me to label very specific parts of the cell in different colours so that I can visualize them under the microscope. The protocol for immunocytochemistry isn't very interesting to watch. It just involves adding different solutions and incubating them for certain time points. And so I wanted to focus more on showing you some of the images I've taken. This is the software for one of the microscopes I use for my PhD. This microscope comes with specialist software which allows us to analyse our images as well as taking the pictures. So if you look up in the top right corner here, you can see that this is a layout of the plate which my cells were grown in, like I just showed you in the previous video. And then the blue circles are the wells which I've actually taken images of. Then, within each well, I can see this big overall picture of the well, which is split up into smaller fields, which you can see by these smaller numbered boxes. I can zoom in on each of these fields to then see a better view of the cells and to do any analysis. So for example, you can see that this number 17 box is in yellow. You might just be able to see that. And if I click here, then it zooms in on that field. And then I can go back and I can click on a different one. Wait a second for it to load. That one becomes yellow. And then I can click here and look at that field instead. So this first experiment I wanted to show you is from a plate of neurons. Neurons are nerve cells in the brain which send and receive messages. They have a cell body, which is these small circular structures, and then they have long projections which branch out to neighbouring cells to allow them to communicate with one another. So all of these long projections in green. In this experiment, I added a compound to some of the wells in the plate and didn't add the compound to others because I wanted to test whether adding it had an effect on the cell. By adding it to some wells and not to others, I could compare the images and see any differences. After treating the cells, I used the immunocytochemistry protocol to label different parts of the cells in two different colours. In green, which I can show on its own, I labelled a protein which is found in these long projections coming out of the cells. And then in yellow, which again I can show on its own, I labelled a different protein called tau, which has an important role in the development of Alzheimer's disease. In these experiments, I wanted to see whether adding the compound affected where in the cell tau was found. So being able to label different parts of the cells with different colours means I can analyse how much the two colours overlap to see where tau is in the cell, and then I can see if it's found in a different place when the cells are treated in different ways. Next, another experiment using neurons. This experiment is slightly different, however, because I added a fluorescent marker into the cells while they were still growing, instead of using immunocytochemistry to label the cells after they were fixed. This means that the whole cell is labelled in green, and you might have noticed that there seems to be a lot less cells in this image than the previous one. This is because this way of labelling cells is a lot less efficient, so not all of the cells on the plate are showing up as fluorescent. If I zoom in here, you can see one of the neurons better. In this experiment, I was interested in finding out whether the compound I used in the previous experiment has an effect on the health of neurons. So in these experiments, I was looking at how complex the structure and shape of the neurons were, because this is a way of seeing how healthy they are. I can also show you how I do the analysis for this experiment. This software is really clever and can pick out the shape of the cells. So you can see here that I run the analysis, and then if you wait a second, it picks out the shape of the cell. And you can see that in yellow, it's picked out all of these long projections that come out of the cell body, which is circled in red. The software measures loads of different parameters relating to the complexity of the shape of the cell. I can then export this data and it gives me an idea of the health of the cells, which is what I'm interested in. Lastly, I wanted to show you some astrocytes as well, which are a different type of cell in the brain, which I also work with. You may have noticed that I said at the beginning that my project is interested in astrocytes, but so far I've only shown you neurons. This is because I'm interested in the effects that astrocytes have on neurons during Alzheimer's disease. In the human brain, these two cell types communicate with one another and work together to do their jobs, and so it's important for me to do experiments with both cell types to try and find out what goes wrong in Alzheimer's disease. So when growing astrocytes instead of neurons, the cells are grown in the same plates and labelled fluorescently in the same way. If I zoom in on one of the fields here, you can see the cells a bit better, and you can see that they're a very different shape to the neurons. And here I've labelled different parts of the cell in two different colours again. I wanted to show you this just to highlight that as researchers we'll use different types of cells to answer different questions. And when researching Alzheimer's disease, it can be helpful to use different types of brain cells to get a fuller picture of what's happening in the human brain. So that was a brief look at some of the ways that I use cells and microscope imaging to answer questions about Alzheimer's disease as part of my PhD. I hope that you enjoyed it, and please feel free to ask any questions in the Q&A. Thank you.
Hello everyone and welcome to the Lab Notes event. My name is Paula and I'm a PhD student here at King's College London, funded by Alzheimer Research UK. I am interested in the role of glial cells, the cells that support and surround neurons in Alzheimer's disease, and particularly my project focuses on understanding the role of a protein expressed in the cell surface of these glial cells that may be important for their immune function and how they trigger responses in front of a neuronal damage or any other change in their local environment. To look into the role of this protein, which is known as P2X7 receptor, and its relevance in Alzheimer's disease, I use post-mortem human brain tissue provided by the Brain Bank, which has been generously donated for research. One of the techniques that I routinely use and help me address questions such as where is this protein located in the brain, or even is there more or less of this protein in comparison to healthy donors by looking under the microscope is called immunohistochemistry or immunofluorescence. And in this video I will guide you through the principle, method, results, and some of data interpretation. So please bear with me. An immunofluorescence is a technique that allows us to detect a particular structure of interest in a slice of tissue. These structures or proteins of interest are going to be recognized using antibodies, which are specific molecules designed to target our protein of interest within the tissue. In turn, these antibodies are labeled with fluorescent particles so that we can observe them under the microscope. But from the moment we receive the tissue from the brain bank to the time we image it, we need to follow various steps. First, the paraffination. This will prepare the tissue for the procedure. Second, blocking, which will prevent our antibodies from recognizing unspecific structures. Third, incubation with what we call primary antibody. This molecule will recognize our particular structure of interest within the tissue. Then there is a secondary antibody, which will come after the first one that we added and will be labeled with a fluorescent probe so that we can visualize our structure. Fourth, mounting. We will add a thin piece of glass on top of the tissue that will prepare it for the final step, imaging, when we will be able to observe the structure of interest under the microscope. Now, I will show you how we actually perform an immunofluorescence following these 6 steps. To prepare the tissue, we will first have to remove the layer of paraffin or wax in which it is embedded. To remove the wax, we'll first place the tissue slides in a rack and perform a series of washes in a colorless liquid named xylene. While this wax is necessary to protect and preserve the tissue before we use it, it needs to be completely removed for an optimal result. Paraffin is also an agent that keeps the tissue dehydrated, and when we remove it, the tissue will need to be rehydrated, using a series of washes, first in ethanol, as we can see right now, and finally in water, here in the blue container. An additional step that we need to add for our tissue to be ready is called antigen retrieval. Once again, the wax has preserved the tissue, but in order to do so, some structures in it could have changed shape. To facilitate that all the structures return to their original conformation, that shape that we find in the human brain, we apply to our tissue heat, immersed in a solution called sodium citrate. Finally, we require a couple of washes in water, here again inside the blue container, and saline solutions that will leave the tissue ready for the rest of the procedure. Some sides of the tissue can be sticky. To prevent the primary antibodies to bind to these unspecific sites, we will first need to add a solution called blocking solution. Here I'm adding a hydrophobic barrier that will contain the liquid solution, and finally I'm adding the blocking solution, which contains proteins that will adhere to these unspecific sites, enhancing our signal and reducing the background noise. Now the wax has been removed from the tissue. All the structures are back to their natural conformation and sticky sites have been blocked. We can finally incubate the tissue with a solution that contains our specific primary antibody. Here I remove the blocking solution and now I am adding the primary antibody. This will recognize our structure or protein of interest. Once the primary antibody has been bound to the target, we can add a secondary antibody that specifically recognizes the primary antibody and is labeled with a fluorescent particle. This fluorescent particle is the reason why we are able to observe a fluorescent colorful signal when we look under the microscope. During aging, the human brain naturally accumulates a compound, namely pofascin, in the shape of fluorescent granules. To dampen down the unspecific autofluorescence of the tissue and allow the specific signal to stand out, we will incubate the tissue with this solution, named Sudan Black, subsequent to the addition of the secondary antibody. The tissue is now stained and just needs to be prepared for the final step, imaging. For this, we will attach a thin glass slide onto our stained tissue together with some mounting medium, which I'm adding right now. The mounting medium is a solution that will help protect our fluorescent signal during the imaging process and also acts as a glue that keeps the glass attached. Here I'm adding the stained sample onto the thin glass covered with mounting medium. Our tissue is now ready for imaging. Here I am using an inverted confocal microscope of the World Cellular Imaging Center downstairs. Because it is an inverted microscope, I will place the tissue slide upside down, which means that the thin glass that was placed during the mounting will be facing the objective. Finally, I will look down the microscope ocular until I see a sharp image, and from this point onwards, I will use the computer attached to the microscope to move around the sample. The imaging program allows us to take pictures of a specific area of interest within the tissue. In turn, as the tissue slide has certain thickness, we could also take pictures across the different layers, creating a three-dimensional image. Finally, here we have some example results. In green, on the left-hand side, we can see a protein called GFAP, or glial fibrillary acid protein, which is a marker of astrocytes. Astrocytes are a type of glial cell with many processes that envelop connections between neurons. They are essential for their communication and support, among many other important functions. In turn, in red, we can observe the receptor I talked to you about earlier in the video, which is the protein I'm researching. By combining these two markers, one in red and one in green, I can study whether this receptor, called P2X7R, is present in astrocytes in the human brain. As we can see in the magnified image on the right-hand side, it seems that P2X7R in red and GFAP in green colocalize, and this is observed as a yellow color, which indicates an overlap. Finally, we can also see how green and red positive cells are found surrounding amyloid beta deposits, here stained in blue. And finally, I would like to thank you all for attending this session. And please do submit any questions you have. I'll be happy to address them in the Q&A.
Thank you. Lovely segue there from Paula into the Q&A. In the interest of time, I'm going to invite everyone in that video that was expertly made to join me for the Q&A. We're lucky enough to have all those involved. So thank you very much, guys, for joining. So we've had loads of pre-submitted questions, some really useful ones, but some really nice live submitted questions as well. And Louisa, I'm going to start with you because I was going to ask you about career advice, basically, for a couple of people that had come to us, and one has come in live. So I'm going to read it out as it is. So it says, I'm hoping to become a dementia researcher, and I found this session really interesting. I'm in the final stages of a psychology BSc, and have applied to the Dementia Neuroscience MSc at UCL. Is there any other courses or routes the researchers would recommend?
Thank you for that question. Yes, so I can't speak for specifically the UCL course, but I know that at King's we also have a neuroscience MSc, and we sometimes in our department, we get those students come and do their their lab experience with us, and we'll kind of supervise them and stuff. And quite often they are psychology students. And I think that is a really good route to go from if you're doing psychology BSc to then trying to then move on to do a neuroscience PhD. Then yeah, like that's a really good route for you, is to do an MSc, because I think just the really key thing is to try and get lab experience. Yeah, that's just the most valuable thing really, is to try and get some experience in the lab, see that you actually enjoy it before you do a PhD, and also just for then interviews and stuff, it's great to be able to show that you've got that.
Thank you. Yeah, hopefully that's a great answer, and hopefully that's helped whoever asked that question. So thank you very much. Paul, I'm going to come to you next, because we saw you in your video doing your thing in the lab, and you know there's been obvious restrictions over the past 12 plus months due to the obvious. Now, one of the questions that's come in is, how has your research and day-to-day work been affected by COVID?
Thank you, Alex, for the question. So basically, so obviously due to the COVID restrictions, also the building had certain capacity limits. We had to reduce the amount of people that could be in the building working at the same time. And that had inevitably an impact on our day-to-day work. Some experiments had to be postponed or reorganized in a way that maybe we were working three full days instead of a whole week, and basically we've had to just rearrange, and unfortunately I'm afraid it has slowed down a little bit the pace of research.
Yeah, thanks. I'm sure it has slowed it down a little bit, but as we saw in the video and listening to you speak, you guys have done a great job in terms of persevering and getting through this. Has been, all of us have had our challenges, but for you guys it must be particularly frustrating. So thanks for answering that one. Jodi, I'm going to come to yourself next. It's a question specifically aimed for you in the Q&A here. So it says, Jodi mentioned that she studies motor neuron disease. Is there a link between this illness and dementia?
Yeah, thank you, Alex. So the type of motor neuron disease that I was looking at in the flies is ALS.
And there's a lot of research in the genes that cause ALS. And actually a lot of these genes have similar mutations that also cause frontal temporal dementia. So we talk a lot about Alzheimer's disease, and that's because it's the most common form of dementia. But there are other diseases that also cause dementia symptoms, and one of those is frontal temporal dementia. So before all of this research, it was thought that ALS was a separate movement disorder, and that dementias are obviously normally memory and cognitive problems. But around a third of people that have ALS actually go on to develop frontal temporal dementia later in life. And so they're very similar in terms of their genetics and their pathology. So if we can study ALS and frontal temporal dementia, we can learn more about how these diseases progress.
Thank you very much. Hopefully that answers the question there. Bea, I'm going to come to you next. Another one that was submitted live that hopefully you can shed a little bit of light on. Now it says, if not everybody develops dementia, even though their brains show the markers for it, does there need to be some sort of trigger, such as a head injury or an environmental factor?
Thank you, Alex. Yeah, that's a very good question. So yeah, there's definitely, and that can be linked to inflammation that we know now that has an impact in the development of Alzheimer's disease. And in fact, there was a huge study that was funded by the NFL in the US, because they showed that a lot of the players that had concussions playing American football showed actually tauopathies, which is a form of that can manifest with dementia. So there is definitely an effect of head injury, and environmental, yes, for sure, inflammation and other factors can affect the disease. Yeah.
Thank you. Yeah, that's really interesting, particularly with the kind of sport link there as well. Just to follow up on that, really, it was a pre-submitted question, and I suppose it kind of links to risk factors. And I'm going to stay with you, Bea, if that's all right. And it's a question we get a lot. And actually, I'll merge the two. Is it possible to have dementia in young age? But also, from that perspective, is Alzheimer's hereditary?
So yeah, this is a question we get asked a lot, especially when you have somebody in your family that is being diagnosed. Actually, Alzheimer's disease is mostly sporadic, which means that there is no mutation linked to the disease, but there are some cases in where it's linked to some changes in the DNA, so that can manifest earlier on, like 50s, 60s. And also there are other forms of dementias linked to other mutations, like frontotemporal dementia can happen due to mutations in other genes that happen very early, like people in their 50s. But it's not as common as sporadic AD, which is the most common form of dementia in general.
Yeah, thanks. Yeah, it's good to have that question answered, because it is one that comes up time and time again, really. So I think often is in the forefront of people's mind when they come to a discussion like this. So thanks for that. So Louisa, a question specifically on your talk. Someone had obviously noticed how you were treating your cells, not in a good or bad way, but it is specifically around which drugs are you using to treat those cells?
Yes. So in the experiments I was showing, it's actually a compound that I was adding to the cells was something called a cytokine, which is like this little compound that cells will release to communicate with each other, and they kind of signal to each other with these slightly different versions of these little cytokines. And basically some previous research in my lab showed that this cytokine is released by astrocytes in the more like disease state, and it releases more of this cytokine in the disease state than it does in the normal state. And so what I wanted to see was if astrocytes are releasing this, is it maybe having an effect on neurons? And so that's why I've been applying it directly to my neurons to see, if it changes the health of those neurons, because that might be something that shows like the mechanism of what's going on in Alzheimer's.
Thank you. Clears that up. Paula, I'm going to come back to you if that's okay. So this is another question we get asked a lot, actually, when people off the back of our talks, whether they're looking to get into research or they want to help take part. Can you point anyone or tell anyone how they can actually get involved in research?
Yes, and thank you, Alex, for asking me this question. I think there are several ways. One of them, for instance, it's through a platform. I think they have a very detailed information that it's called Join Dementia Research. And this is a platform in which every member of the public could get involved in research. They can get involved in different types of studies, ranging from prevention of dementia, some questionnaires, to help address the well-being of people living with dementia, but also the carers, and also some therapeutic studies, if I'm not mistaken, clinical trials, but also this is a great platform, and there are many other ways, such as, I don't know, maybe providing funds to Alzheimer's Research UK or other dementia organizations, or even, of course, developing a career in dementias, like all the other panelists here. There are many ways to access this.
Oh, thank you. And yeah, nice mention of Join Dementia Research there. I'm sure towards the end of this event, we can probably get a link out to Join Dementia Research website, actually, because it's a really useful tool for people to use if they want to get involved. So that's great. Thank you for that. Now, this was a generic question that came in to everyone, but obviously time constraints, we have them. But so Jodi, I'm going to come to you. And it's simply put, why did you choose the particular research that you undertake?
Thank you, Alex. Yeah, I get asked this quite a bit, because obviously it takes up a lot of your time doing a PhD, so you have to really love it. And if you don't, then you're not going to enjoy what you're doing. You're not going to be good at what you do. So I actually worked as a nursing assistant before I decided to pursue neuroscience. So I worked with dementia patients and with their families, and I saw that the way it affects them day to day. I think it's different kind of reading about it and hearing about it, seeing it on movies and things like that, versus actually seeing it and experience it firsthand. And so that made me want to kind of navigate a career that meant that I could make a difference in those spaces, and that's what I'm hoping to do.
Yeah, it's key, isn't it? That you've got to love what you do, especially if the level of dedication that you guys have to kind of commit to your work has to be key to obviously enjoy that. Now, Bea, I'm going to come to you with what sounds like a question that I'm presuming you've been in the game a long time, so don't take offense. But the question is actually, in the last 25 years, so I'm not suggesting you've been working in this field for 25 years, but what have you learned about Alzheimer's?
Thanks for the question. At least I was born, not like the other panelists. I was born 30 years ago. But yeah, I think it might seem like we haven't progressed a lot because there is actually no treatment for Alzheimer's disease. But Alzheimer's disease is a very, very complex disease. And also the brain is a very difficult organ to access with drugs. So it might seem that we haven't learned a lot, but I think we have learned a lot since Alzheimer's described the pathology. And what we thought in the beginning that it was a very linear disease, you have a beta, then tau, then neurodegeneration. We know now that it's not really like that, but it took us a lot of years of research to overcome that hypothesis. But that's how science works. You have one hypothesis and you have to test it. So now we know that there are a lot of factors that are relevant to develop dementia and Alzheimer's disease besides amyloid beta and plaques and tangles. So we know that. And also the disease manifests much earlier than the symptoms. So once you show like you don't remember things or you get lost, the disease has been there for a long time. So now we know that it's very, very crucial to diagnose the disease earlier. And we have a very good new set of biomarkers, new neuroimaging techniques. So we are able to identify patients earlier on, and also to discriminate patients, like they are all not everyone is the same, and we should focus on a more personalized medicine. And I'm optimistic about the future, but it took us very long to get here, so it's a bit sad at times. Yeah.
Yeah, well, I'm going to focus on the optimism. I think that's a nice way to close out the Q&A. And with that said, I just want to thank you all for not only being here for the Q&A, but for the effort that you've put in with the videos. I know things have been really busy and you've been under certain conditions of COVID. So yeah, thank you all for A, attending, but B, putting the effort into the videos. So yeah, on behalf of the charity and myself, thank you.
Thanks for having us, Alex. Thanks, everyone.
Yeah,
Thank you very
thanks
much. Sorry.
No,
Thanks.
I just wanted to thank you for having us, and that it's been a pleasure joining the session.
Thank you, and I would like to say thank you to Alzheimer's Research UK, because actually a lot of this research has been funded by them, because I was a fellow. Paula is a PhD student with them, so thank you very much.
Yeah, thank you so much for having us. It's been really nice to answer all your questions and see what you're interested in.
Cool, great. So as we just go over the other side of midday, I want to give my huge thanks once again to the speakers for sharing what work in the lab is all about and for answering loads of questions there in the Q&A. We've got a couple more poll questions if you can stick with us for another minute or so that we'd appreciate you answering. Again, these are helpful as they give us an insight into how you found today. So I'll give you a few seconds to answer those. Cool, so hopefully you've had the opportunity there to fill those in. And I'll just check out the answers to see whether you've enjoyed, which you have. Brilliant stuff. So the vast majority of you plan on attending further events, which is great. This is why we do it. We want to prolong these events online as well as get the in-person events up and running again. So I'm really happy that you found it useful today. So thank you very much. We'll be sending out another feedback survey, a more detailed one, and that will be along with the video recording of this event in the next few days. So please complete the survey, as we appreciate all the feedback, because we're looking to continuously improve. Now, the next event in the series is on the 13th of May, and that's at 2 p.m. until 3 p.m. That's where you can hear from our North Research Network. And they will discuss research underway at Newcastle University to better understand dementia with Lewy bodies, which is the third most common type of dementia. So do check out the website for more details and register there. If you want to find out more about leaving a gift in your will, as we referenced earlier, please check out the resources and information on our website. If you have any questions about dementia and research that we weren't able to answer today, then please get in contact with our Dementia Research Info line. They can answer all your questions and signpost you to sources of information and support. And as we close out, I want to say a huge thank you for attending. I hope you've enjoyed the event and you've learned something new. Thank you for your ongoing support for our work, and I hope you can join us for another Lab Notes event soon. Thank you.